Hi, good evening. Hope I'm audible. Am I audible? Okay. I'll share my screen now. Okay. So, yes, good evening, all. Welcome to the live practice session for the NPTEL course Applied Environmental Microbiology. I'm Guru Priya, a Prime Minister's Research Fellow in the Department of Civil Engineering, IIT Madras. So, in the week 8, the contents were about water and wastewater microbiology, built microbiology, and about exposomes. So, before starting on with today's session, do you have any doubts from the previous weeks? Any concept wise or question wise? Any doubts? Okay, I guess there are no doubts. And if you want me to discuss any particular topic from any of the weeks or from any topic from this week's course content, you can let me know through the chat box or through unmuting yourself. As of today, I'll just display the first question for today, and then, as always, we'll discuss about that concept by solving some questions. So, yes, the first question for today. Okay, right. So the answer is D. Um, can you just name some physical treatment methods that are used for wastewater treatment? Sedimentation, filtration. Great. Some chemical methods. Chlorination. Okay. Anything else? Ionic exchange. Chemical disinfection. Okay, right. So you all are aware about few physical and chemical methods that are used for water and wastewater treatment. So where do we generally use physical treatment? Like generally wastewater treatment, it has three stages, right? I think you are aware about the primary treatment, the secondary treatment, and the tertiary treatment, right? So in that sense, where do we use these physical mostly? Was the question clear? We have three stages, right? Primary treatment, secondary treatment, and tertiary treatment in wastewater. This physical treatment methods, in which stage is it predominantly used? Primary, right? So, what do we do in the primary stage for which we use this physical treatment? Okay. Okay, we use sedimentation. Apart from that, do we use anything else? No, we don't use coagulation. We don't use it very often. I'm just asking about the physical treatment techniques. If you go for coagulation, you will be adding some coagulant, so it will not be a complete physical process. It will be a physicochemical process. Okay. Let me just give you an overview of this treatment processes and like different things that we use in it. I'll just use my pen now. So, okay. So, generally in wastewater treatment, what we have is we have primary, secondary, and sorry, tertiary. Okay. So, in primary, what we do is we remove easily separable solids. Okay, easily separable solids in a sense, just imagine, for example, we are treating a municipal wastewater. Okay, when you are going to have a municipal wastewater flowing into a treatment plant, you will be able to see many suspended particles in it. Suspended in the sense you can see things like plastic papers or something, plastic covers in it, even floating in it. So right from that to other smaller easily separable solids, all of them you have to be able to separate it out first itself. Otherwise it will be a big hindrance for the other two processes. So what you do here is, in a primary, you go for separating easily separable solids. Okay, so easily separable solids only you are going to separate. In this, generally in the treatment plants, what we use is we either use a grit chamber, we use screens, we use primary settling tanks, etc. So when it's going to be a grit chamber, again it'll have a openings in it, so you can have your sediment settled or retained in the grid chamber. Screens, why do we use? Because when you are going to pass through the screens, all the objects that are greater than the screen size are going to be retained again. In the primary sedimentation tank, what you do is these suspended particles generally get settled in that, and from the primary sedimentation tank you take your effluent to the secondary treatment. Secondary treatment, what we do is we generally remove organic matter. Okay, we go for organic matter removal. The organic matter can be dissolved or it can be colloidal, but we remove organics in the secondary treatment. And uh, mostly we go for biological treatment here. Okay. Secondary treatment is mostly biological, like activated sludge process or some anaerobic process. Many such biological processes are there. So you will go to one of those processes. And in the third stage, you are going to remove nutrients like you're going to remove the other nutrients that are present, and also you will be removing the residual pollutants that are present. Okay, residual pollutants, and sometimes you will have some metals or something which is also again harmful if we are going to consume it. So you are going to remove all of it. So in this primary, secondary, and tertiary, the primary mostly uses only physical processes or unit operations. Okay, whereas the secondary and tertiary treatment will use unit processes. So in secondary, as I told, you will have much of biological processes, and tertiary will be some physico-chemical processes. Okay, physico-chemical processes in the sense you have this ion exchange, you have what it is, adsorption, etc. Okay, coagulation. So all of it you will be using in your tertiary treatment. So do you have an idea of to what level we have to treat this? Is it like, do we have a same standard for all a treatment plant? We give some input value. So what we will check to tell that okay, this water is treated? What are all should be checked, or the parameters basically? BOD. Okay. Anything else? BOD is right. COD. Okay. You're studying about microbiology, right? So based on that, what parameter will you check? If the BOD and COD is fine, only that's it. Initially, we studied about MPN test, plating, and all. Before ARG, you have to check something very basic. Pathogens, right? So you call it as total coliform and fecal coliform. Okay, total coliform and fecal coliform. You call it in the terms of like water waste parameters of water testing. You call it as total coliform test and fecal coliform test. Okay, as you can know, total coliform is the total organisms that are present, and fecal coliform, E. coli or something like that. That is again an indicator of contamination in the water. So generally, you take a water sample and you're going going to analyze various parameters. See, again, like the treatment methods, the parameters can also be physical, chemical, and biological. Uh, in order to certify that this water is safe to drink, you have to have all those parameters under specific value. So there are many standards for this value, so you can follow those standards. But at end of your treatment, all those values should be less than what is allowed. Okay, if there is, like, we can say only 5 milligram per liter of something is allowed, your after your treatment it should be under 5 milligram per liter, then only that water can be certified as treated for that purpose. And one important thing to note here is the purpose also depends. Drinking water standards may not essentially be the same as the water standards for some other purpose. For example, you're going to use your water for watering crops in the agricultural land. The testing or the parameter value range will be something else. For drinking water it will be something else. Just for recycle and using it for flushing purpose or some other industrial purpose, in industry also, for what purpose you are using, like if you are using just for a cooling purpose or heating purpose, in that case your water can have some different standards. In some other case it can have different standards. So whenever you are going to think about wastewater treatment, just have in mind that your final wastewater should have a characteristic parameter that is lesser than the standards mentioned for the purpose for which you are going to use the water. Is that okay? So this is very basic about wastewater treatment. Is that fine? um, should I also give you some examples of what different characteristics you have to be checking? Just give a reply in the chat box. Okay. Okay, so as I told you, the characteristics of wastewater can be physical, chemical, or biological. So, in physical characteristics, you will be checking things like the color of the water. See, color basically indicates the presence of pollutants in wastewater. Also, a good appealing color is very important when you are going to supply a wastewater. Aesthetically, it is also very important. Both technically and aesthetically, color is a very important factor. Apart from that, we have order, we have turbidity. The water's turbidity is very important. Temperature, pH, and solids content in the water, all these come under physical characteristics. Okay, and in the case of chemical characteristics, as you people told, it was BOD, COD. You will also check about the total organic carbon, other nutrients in it, like how much nitrogen, phosphorus, or other metals, heavy metals, toxic chemicals, hardness, alkalinity of the water. All of this you will be checking in the chemical characteristics. In the biological characteristics, you will be checking about this fecal and total coliform, this ARG. Based on your need, you will be checking the different biological characteristics. Is that fine? Can we move on to the next question, or you have any doubt in it? If you want me to repeat, you can just post repeat in the chat box, or you can give a okay or a thumbs up so that I'll go to the next question. Right. So Right, it is B. Obviously, microorganisms are going to decompose organic matter. We use microbes for removal of nitrogen and phosphorus as well as heavy metals, and they only don't do filtration of solids. This is your next question based on whatever we have discussed so far. Okay, so grid chamber is used for removal of sand and gravel. Extended aeration tank works well for low strength sewage. So, what is an extended aeration tank? Two people have answered it right. You've answered C right. Yeah, grid chamber, as we discussed, is used for removal of sand and gravel. It's a primary treatment and a physical process. Completely fine. Extended aeration tank works well for low strength sewage. You can you can just give me what an extended aeration tank is. And why it works well for low strength sewage, if you know it. Sewage treatment method. Yes, obviously it's a sewage treatment method used activated sludge. Okay. We have activated sludge, so why do we go for extended aeration? No, aerobic bacteria digesting organic matter. Yeah, it's partly right. Even if you can't type, you can unmute and answer. In ASP is aerobic process or anaerobic process. ASP is a aerobic process, right? So what is the difference? Have you studied about ASP and things in detail? I mean, was it discussed somewhere, or have you read or known about it? Okay, fine. So generally, we call something as a treatment called activated sludge process. Okay, in activated sludge process, what you have is, after your primary treatment, you will have one tank known as the aeration tank. So in that, aeration will be provided, obviously. So what happens is, the when the aeration is provided, all this aerobic bacteria will consume upon the organic matter that is present in the water. The soluble organic matter can be easily uptaken, right? So we have two things called soluble organic matter and colloidal organic matter. That is just for your information. Now just remember, we are completely talking about only the soluble matter. Okay, the soluble matter can be easily taken up by the microorganism, because it has a lesser surface area and the diffusion can be better. Okay, so it takes the organic matter, it grows, the cell grows, the cell uses it for its growth, so the cell size also grows. And finally what happens is, at a point, the all the organic matter is being consumed, so now the water is uh, doesn't have any soluble organic matter, but again the water contains all our microorganisms which are now grown. Okay, so that is also a organic matter. At the end of the day, when you're going to consider what are all organic matter present, now our soluble matter has converted into the colloidal matter, which is our cell. So what we do is, after the aeration tank, we send it to a secondary sedimentation tank, where the sedimentation happens and all our microbes are going to settle down, and at the top part you will have oops. Am I audible? Hello, am I audible?
Yes, ma'am.
Okay, okay, fine. I got disconnected for a moment. So what happens is, after this growth and all, it goes to the secondary sedimentation tank, where your cell gets settled down, and the top part will be your treated water, which you will can take to your tertiary treatment, or you can just use it if the standards are fine. Okay, this is the general ASP process. So in ASP process, we do some process modifications, basically, with regards to the aeration. Because like, the the process is not as theoretical as we study it in ASP. In real time, we have many pros and cons, and many challenges with the process. So what we do is, we go for an extended aeration system. In one case, there are many modifications there, but one modification is going out for an extended aeration. As the name suggests, you are going to produce an extended aeration for the system. So what happens is, when you're going to have a low strength wastewater, the food is to biomass ratio will be low. Okay, so I hope you are all aware about what is the food is to biomass ratio. Food is to biomass ratio is whatever is this BOD of the water, or the organic that is present in the wastewater to be treated, and the microorganism that is present in the water. This is called the F by M ratio, or the food is to microbe ratio. When this is low and you are going to give an extended aeration, what happens is, the microbe tend to stay in a stationary phase for a longer time. Here you will take a longer time, as the name suggests, it is an extended process. Okay, so extended aeration you are going to provide, with higher retention time in the reactor. So with that, what happens is, your microbe stays in the stationary phase, which is the endogenous growth phase, for a longer time, and it can like very well work for a degradation of the low strength organic matter in that condition. Okay, there are many other modifications which are used for different applications. So one of it is extended aeration tank, where in the extended aeration, you will generally maintain a low food is to biomass ratio and provide aeration for a longer period of time. You are going to have the treatment time a bit longer. So what happens is the cell is going to stay in a stationary phase of growth, and the removal of organic matter happens. Is that clear about extended aeration? Okay. Is any doubts? Okay, I assume there are no doubts. And the primary sedimentation tank. Primary sedimentation tank to remove the suspended solids. Tertiary treatment is to remove nitrogen and phosphorus. I guess you are clear with this. The only catch here in this question was about extended aeration tank and the low strength sewage. Low strength sewage means the organic content or the matter is going to be low in a very common term. That's a low strength sewage. And as I explained, that is the reason why we go for external aeration for our low strength sewage system. So here is the next question. Okay, before you answer this question, let me just ask you, what is BOD? You can just give me the full form and also what it means.
Biochemical oxygen demand.
Yeah, right. Perfectly right. What does that indicate?
The oxygen dissolved in the wastewater that is available for the microbes to feed on.
Can you please repeat?
It is the oxygen dissolved in the water which is available for the microbes.
Okay. Is it the oxygen dissolved? Any other answers from anyone else?
It is the oxygen required by the biological organisms to consume the waste in the water.
Okay, right. Right. I think you are clear now. It is the amount of dissolved oxygen that is needed for a complete breakdown of organic matter in the wastewater by microorganisms. Is it clear? Is it clear to everyone else what a BOD is? A BOD is nothing but a biochemical oxygen demand, which means how much oxygen is required for complete oxidation of the organic matter using biochemical route, that is by using microorganisms. And similarly, COD is nothing but chemical oxygen demand, where it is the same oxygen demand for complete oxidation by oxidation of the organic matter in the water by chemical methods. Okay, so that is the difference between COD and BOD. So now, what is BOD 5? Very simple question, right? What is BOD 5? incubation after 5 days. Right. There are a lot more details about a BOD test and the method of calculating BOD. If you are interested in knowing it, maybe you can respond. I think there are some three people here around. If two of you want me to explain, I'll explain it. Otherwise, you can just go on proceeding with solving this particular problem. Okay, sure. I guess one of you want me to explain. So I'll explain this. You just solve this problem, give me the answer. When after few of you give the answer, along with the answer, I'll discuss about a BOD test and other details of. It's a very simple problem, right? If you are able to solve it, you solve it. Otherwise, please let me know in the chat box if you're not able to solve, so that I won't wait for you to solve it. Think we are just three or four people here, so okay. So I'll explain it now. See, generally we do a BOD test by finding out the dissolved oxygen. Uh, it's not very important for you, but just to tell something, just to give a complete explanation, I am just telling you how to do a BOD test. Okay. It's just for extra information. You need not like take this one very seriously. You can just note it down for your reference, but don't take it very seriously in the view of this course exam or markings. Okay. So what you do is for BOD experiments, you take the sample and you will perform dilution of the sample. See, generally, you have something called a BOD bottle, which will hold around 300 ml only of the sample. Okay, so what you have to do is when you have a wastewater, you have to dilute the sample accordingly. There is some procedure for dilution which is like very advanced level, that is not required for you now. You just remember that you have to dilute your sample using some nutrient solution or tap water or something like that, and then take the diluted sample and add alkaline iodide sodium acid. Okay, and then you have to add concentrated sulfuric acid. You then have to do sodium thiosulfate titration. Okay. uh, when you do the titration, you have to add starch and you have to again titrate under until it is colorless. So this is all you have to do. This is the experimental part of it. When you are coming for your dissolved oxygen calculation, what you will do is you have done a titration experiment basically. I hope you understand. You have done a titration experiment. So what you do is you just have a formula called dissolved oxygen is equal to volume of the titrant or the sodium thiosulfate solution that you had into its normality into 8000 divided by sample volume. So this is how you will find out this DO. So for you what it is given after 5 days of incubation at 20 degrees Celsius, initial and final dissolved oxygen concentration were 9 and 3 milligram per liter. So I with whatever procedure and formula I told till now, you are just doing only one thing, you are finding out the dissolved oxygen concentration. So first you will take the sample, dilute it, and find the dissolved oxygen concentration using the titration method. That will be this. You will allow that bottle, you have to tighten up the bottle very well such that there is no air going inside the bottle. Leave it for 5 days aside. After 5 days you come again and do the same titration procedure and check the BOD after 5 days. Okay, I mean sorry, the dissolved oxygen concentration after 5 days. So that is around 3 milligram per liter. Now what they have asked you to find is what is the uh, now from that what you have to find what is the BOD of the wastewater. In this case you have incubated it for 5 days, so it is the BOD 5 of the wastewater. So that how will you find means like dissolved oxygen initial minus dissolved oxygen final by the dilution factor. Okay, that is the formula. Dissolved oxygen initial minus dissolved oxygen final by the dilution factor will give you the BOD, and this will be in terms of milligram per liter again. Is that clear what a BOD test is?
Yes, ma'am.
Okay. One more explanation is generally in all parts of the world we have this habit of finding out the BOD after 5 days. That is because of the fluctuations in the dissolved oxygen concentration. Because like when you're going to leave it for so many days, sometimes it may happen that you are going to seal the bottle tightly, so all your dissolved oxygen may get exhausted. That is all, that will also show you a errored result. And also when you do it in 1 day or 2 days, you may not get the complete oxidation, so that will also give you a error prone result. So that is the reason optimally you take it for BOD 5 at 20 degrees Celsius. Okay, but in our country and all, maybe this kinetics may be a bit faster, so you can go for something called a BOD 3. In somewhat colder countries, you know that when it is going to be a colder country, the microbial activity is going to slow down because of the lesser temperature. When you're going to increase the temperature, the microbial kinetics and the reactions will be at a faster rate. So maybe in our country we can take this as BOD 3. It's not a very standard thing that you have to have it like this, and you can have multiple values also, BOD 1, BOD 2 to BOD 10. All this you can have. Okay, so I hope it was clear, and with whatever I told, can you now give me the answer for this question?
Ma'am, C.
Okay, I have got one C. Okay, two people telling C and one A. How is it A? Okay, anyways, answer is C because initial value is 9 minus final value is 3, which is 6, divided by the dilution factor. Dilution you have done it to 1 is to 5. So 1 by 5. So it will be 6 into 5, 30 milligram per liter. So for your exams, And also one more possibility is this exact formula is going to be DO initial of minus DO final of the sample minus DO initial of blank and minus DO 5 of blank divided by dilution factor. So in case any value for blank is given here, you have to subtract it from your sample value. That you do in any other experiment also. Basically, I think you all are familiar with the spectrophotometric experiment. So when you're having some other blank value, you set it to 0. Otherwise, what you do, you tend to subtract the blank value from your sample value, right? So then the similar way, if there are some DO concentrations given for blank also in your question, just subtract it from the value of your sample DO. Have these concepts in mind. You may get questions of this sort for this exam of this paper. Okay, so you can answer this question now. Very simple question. I think it's beating out the bush again. You all are right. What are persistent contaminants?
Some heavy metals.
Okay, that is an example of persistent contaminant.
Ma'am, the contaminants that cannot be removed by biological or physical means.
Okay, DDT. Okay, all these are examples. So persistent contaminants are the contaminants which have longer half-lives. So generally they are persistent, means they stay there for a longer duration of time. Like degrading them is a problem right there. If it's going to be organic matter, they are not persistent because you can easily degrade them. You can have biological methods, you can have chemical methods. You're going to easily degrade them. But degrading a persistent pollutant is very difficult. And also we have something called a POP, which is a persistent organic pollutant again. So not all organic pollutants are even, like, not all the time they are easily removable, something like that. Okay. uh, okay, I guess we will also be discussing about emerging contaminants. Let me just introduce it now. No issues. What are emerging contaminants then? We have something called persistent contaminants. Okay, they have longer half life, difficult to degrade. We cannot go for biological treatment for them. You gave examples also. All of that is fine. Now, what is emerging contaminant or contaminants of emerging concern? Okay, assuming that you have no idea about it because I don't get any response, I'll just tell you. Emerging contaminants, as the name suggests, are contaminants, like they're not very new contaminants and all. It's just that we have not so far recognized them as contaminants or something. Okay. Um, I'll tell you with an example. For example, you have a contaminant. It is present in the water. It is affecting you. It is affecting the human health, other as well as other organisms' health. Maybe in the past, some 2, 3 years or 5 years. Okay. So now you know there is some issue. When you're going to test the water, you find a commonality of the presence of this one particular pollutant in the water. Okay, so now you know there is a correlation between whatever the harm that it has caused and the particular pollutant or the compound, but you have no evidence of it. I mean, the research is in a very nascent stage where you don't have very strong evidences of what pathway it is going to go and affect. In, for example, a fish, for example the humans, we know there is effect, but we exactly don't know what is the toxicity mechanism that's happening right there, or you don't know under what concentration even it is that is toxic. See, we all know metals are toxic, but even in our water we have something called a permissible limit for everything. So permissible limit is something up to which our body or uh, our body can accept that. Above that it is going to turn toxic and harmful for us. Okay, that is known as permissible limit. But for this emerging contaminants, the problem is you don't know what is this permissible limit is. You are just still studying it. There are no standards for it because it's all evolving right now, and researchers are studying about under what dose it is toxic, and all. So based on the study results, when we get a standardized results only, we will again go for setting up standards for that particular compound, right? So these are known as emerging contaminants or contaminants of emerging concerns. You can take some antibiotics maybe, or you can take what, something called endocrine disrupting chemicals are there, like plastic based chemicals, like many plastic based chemicals are there which can act as potential endocrine disruptors, which means that can disrupt your endocrine system basically, not only ours but also other organisms also. It can affect the endocrine systems basically. So that is very deadly, right? But we don't know under what concentration it is going to do, or what is the potential effect that don't have a complete study or complete understanding of that particular pollutant. Those pollutants are known as emerging pollutants, which is a very hot area of research in terms of this um, field is concerned. Okay, so that is the difference between persistent contaminants and emerging contaminants. There are many more terms related to this area of research or this field basically. So when you read something new, just go back and see what that particular term means. You have to have a clear idea of what a particular term means and what are the examples. So when I ask you what it means, just don't give me examples. Yeah, you also have to give me, but along with the definition if you give me, then I will have a clear understanding of, okay, you have understood or you have known the term really very well. Because this area you have many new and similar terms which can like sound similar, but obviously they have their own importance. So that is why I'm explaining even all the options in one question. Okay, so with that we'll go to the next question. Okay, right. Okay. Oh. So A is the right answer. Uh, why do we focus more? So we have told about nutrient removal in the tertiary system, all those stuff. Why are we more concerned about nitrogen and phosphorus basically?
Ma'am, it is present more in the environment.
Okay, right. It is present more in the environment. And one more thing is they potentially can cause eutrophication. So two main nutrients in that are involved in the process of eutrophication are nitrogen and phosphorus. I hope you all know what eutrophication is. It's just basically the algal bloom, and that's going to disrupt the entire water ecosystem. That's very deadly. So that is why we go for this nitrogen and phosphorus removal. So in the phosphorus removal, as far as this particular question is concerned, all other options are uh, like totally disconnected to whatever we have asked. So that is why one way of saying the answer is that is why option A is the answer. But to discuss upon all these options, first let's start with the other options only. Nitrosomonas and nitrobacter, what are they used for? Okay, let me tell you. Nitrosomonas and nitrobacter are used in the process of nitrification. So in nitrification, what happens is when you have ammonium in your water, it is first converted into nitrite. So nitrosomonas basically aids this conversion of ammonium into nitrite, and this nitrite is further converted or oxidized to nitrate, and this conversion is aided by nitrobacter. So that's the role of nitrosomonas and nitrobacter. Obviously, when nitrification, only denitrification is going to remove the nitrogen from our system. So nitrification, what happens? Ammonia to nitrate is called nitrification, right? Whereas in denitrification, again from this nitrate, you will go back to get N2 nitrogen, which will be one method of removing the nitrogen from the water system. Like N2 is a gas, so obviously it will evolve out. So this is known as nitrification and denitrification. So I think you are clear with option B and C. What are acidogenic and methanogenic acidogenic bacteria and methanogenic archaea? Also, I hope you understood what I told about B and C. If you want any clarifications, let me know in the chat box. Also, answer those. Yes.
Ma'am, can you repeat about nitrobacter?
Okay. See, your nitrification has two steps. Basically, NH4 plus to nitrite, that is NO2 minus. Okay, so that step is aided by or the metabolism happens by nitrosomonas bacter. And the next step is like this nitrite is very short lived. Okay, it is very short lived. This NO2 minus is converted to nitrate, which is NO3 minus. This is aided by this conversion is aided by nitrobacter. So nitrosomonas and nitrobacter are basically nitrifying bacteria. Is that clear?
Yes, ma'am.
Okay. So after nitrification, this denitrification process generally takes place, where again in the form of N2, like your nitrogen will be removed from the system as N2. your nitrate NO3 minus will be converted to N2. This is known as denitrification process. So that is what the explanation about B and C. Now I'm asking you about D, option D. What is acidogenic bacteria? What is a methanogenic archaea? Where do we hear all these names and terms? No idea? Okay, if you don't have any idea, just give me a thumbs up so that I know you have no idea and start explaining. That's it, very simple. Okay. I hope you all know about, you have heard about anaerobic digestion, just the term anaerobic digestion. So, till now we have been discussing about ASP, aerobic process, nitrification, denitrification, phosphorus removal, all of it. There is one more biological process called anaerobic digestion. So, in anaerobic digestion, you have several stages. Okay, like it takes place in four stages. A very basic thing is it happens in four stages: hydrolysis, acetogenesis, and methanogenesis. Okay, hydrolysis means you have an organic substrate, like I um, I just give you complex polymers, like take it, be it carbohydrate, proteins, or lipids. I give you complex polymers. So for the bacteria to metabolize it, first step is to hydrolyze into utilizable things. Okay, so it will like convert it into simple sugars, amino acids, etc. So this process is known as hydrolysis. And then acidogenesis, in a sense, using all those hydrolyzed products, your bacteria will start producing volatile fatty acids. Okay, this is known as acidogenesis step. And the third step is acetogenesis. Acetogenesis, then acetic acid is produced basically. And then for this, all these steps are like uh, happens one after the other, they are sequential. So after this acetogenesis step, until this bacteria will have its role. So until this step, but in the next step for the methanogenesis, what happens is there are methanogenic archaea, where the archaeal organisms will come into the picture. They will use different things. For example, they will even use the acetate that was produced and convert it into methane. So they are going to generate methane. So henceforth, they are known as methanogenic archaea. So, acidogenic bacteria are the bacteria that are going to produce acids, and methanogenic archaea are archaeal organisms that are going to produce methane. Okay, so this acidogenic bacteria and methanogenic archaea come into picture in anaerobic digestion process.
Ma'am, so they are they in a symbiotic or some kind of relationship?
Yeah, they have syntrophic relationship. I guess in one of the classes I explained you about syntropism. So they have a syntropic relationship. Without acidogenic bacteria producing acid, the next step cannot proceed, and so the sequence will not happen even if one of the step is disrupted. So one product, the product of one of the organism is going to be the substrate for the next set of organism. So that is why this sequential relationship is known as syntrophic relationship, syntrophism. In symbiosis you will just have mutual relationship kind of a thing, but here what you have is a sequential relationship. One happens only if the previous thing is going to happen perfectly without any unwanted changes or errors. So that is why this is called as a syntropic relationship. Okay, And coming to the phosphorus accumulating organism and glycogen accumulating organism, um, I think polyphosphate accumulating organism, you all know PAOs. They are obviously going to be involved in phosphorus removal process. Where do these glycogen accumulating organisms come into the picture? Any idea? Okay, let me just give you a general explanation. This the tendency of this phosphorus accumulating organism is they tend to store polymers like PHA, PHB, etc. Uh, okay, they have this intracellular polyphosphate and glycogen. They have both this phosphate stored as well as the glycogen stored. But what happens is under certain conditions they tend to store phosphorus, under certain conditions they tend to release the phosphorus depending on their metabolic needs. They do this. Okay, uh, whereas this glycogen accumulating organisms are going to be in competition with this phosphorus accumulating organism. The competition with the competing factor between this PAO and the GAO is the carbon source. Okay, I told both of them have the tendency of accumulating PHA and PHP, polyhydroxyalkanovate and polyhydroxybutyrate, all these polymers, they have this tendency of accumulating them. In case of phosphorus removal bacteria, this shuttle is mediated with phosphorus intake and phosphorus removal, whereas in glycogen accumulating bacteria, the storage of this PHA and PHB is in trade with this glycogen replenishment. So they are going to basically compete for this PHA PHB storage or the carbon source that is present in the system, water system. So when your GAO is present, your PAO is going to get affected. So that's the relationship between PAO and GAO basically. And both of these are involved in the biological phosphorus removal process. Is this question clear? Give me a thumbs up if this question is clear so that we'll move on to the next question. Okay, this is the next question. A quick question, just give me what is EBPR before you answer this question.
Ma'am, enhanced biological phosphorus removal.
Right. EBPR stands for Enhanced Biological Phosphorus Removal. Okay, as one person has answered it rightly, the answer is D. So obviously we know it is not going to promote nitrifying bacteria or denitrifying bacteria. The confusion will be between it promotes the growth of polyphosphate accumulating organisms, and phosphorus accumulating organisms assimilate volatile fatty acid and produce hydroxybutyrate storage products. So what happens is, in the anaerobic zone generally, the phosphorus accumulating organisms tend to assimilate products like PHB, which I told, polyhydroxybutyrate. When you are going to again bring those from the anaerobic zone, so once they have accumulated this PHB and you are going to take them to aerobic or anoxic zone, what they tend to do is they tend to take up the phosphate that is present in the system. So by this way, the PHP will be utilized, but instead of that, phosphate will be stored in the organism, and this is the method by which the phosphorus being removed from the system. So this is a very sensitive process. When you are going to play with this oxygen availability in the system, it is going to decide whether phosphorus is going to get accumulated, or phosphorus is accumulated in the bacterial system or by uptake, or whether it is going to be released into the system. So instead of removing phosphorus, sometimes it happens that when you are going to give extended aeration, or when you're going to leave it for a long time in the presence of air and all, again the phosphorus may tend to come to the system itself. Okay. So this is a very sensitive process here. Anaerobic, so that's what I told, like you're going to give extended aeration and leave it for more time. What happens is this, there is chance of this reversal happening. So in the anaerobic zone, what happens is phosphorus accumulating organism assimilate VFAs and produce PHB storage products. This oxygen relevance and the individual steps that take place in the process are more relevant. So that is why D will be the answer for this particular question. Yeah, you've answered it right. Genetic factors is not a part of exposome. What is an exposome? What is an exposome? Okay, exposome is nothing but all the exposure for a particular organism. So why this exposure is very important is that this exposure has a significant effect on the organism and its lifestyle and several other factors involved with the survival of the organism. So we are studying about all that an organism is getting exposed to. So in that sense, they may be exposed to chemicals, they may be exposed to other organisms or other enzymes or other biological factors, and also physical stressors like pressure, heat, etc. But these genetic factors are not something that the organism is getting exposed to, but it is present in itself. So that is not a part of the exposure of the organism. One more question. Okay, so two answers we have got. What are biomarkers? Again, as it is stated in statement A itself, biomarkers are some biological molecules. They are nothing but some biomolecules, maybe a protein or something else that is present in the um, blood or other body fluids or tissues. Okay, they will be indicating if there is normal condition present or if there is any abnormality present in the system. And the statement B states that by measuring the levels of the biomarkers, researchers can assess the extent of exposure to environmental factors and evaluate the risk of developing a disease. So it's not only that only when we develop a disease, only when the disease is developed, we get a biomarker. There are few biomarkers which can tell us, okay, we are in an um, state to be warned where we can develop the particular disease. So even that is possible. All these are estimated by knowing the biomolecules that are present in the system in the normal condition. There is some variation, we can tell that okay, this abnormality is going to lead to this particular result. So based on that, both A and B are true. Again, follow up from the first week. I hope all of you will be able to answer this. All right, proteins are building, amino acids are building block of proteins. Here, if you note carefully, all these antibiotic pigments and terpenoids are may fall under the secondary metabolite category. So we'll just discuss a few terms for today and we'll just wind up the class. What is crown corrosion? Just give me answers quickly. Crown corrosion, where does it happen? What is it? Have you heard about crown corrosion or no? Okay. So generally we have a pipeline. So we are collecting water for wastewater treatment and all to sewers, right? So we have the sewerage system. Sewerage system is nothing but the pipeline system. Okay, so this pipeline system is not, you are not going to have water to the fullest level of the pipe all the time. You just have a flow and there is going to be some head space in the pipe above the water level. Uh, Two things are being told. One is like we have this sulfur content in the water. Okay, we have this sulfur oxidizing bacteria or sulfur reducing bacteria. We have various kinds of sulfur utilizing bacteria basically. So what happens is sometimes this H2S gas is produced by the reactions in the water because of the microbes. So this H2S hydrogen sulfide gas is a corrosive gas. This gas, when it's going to be built up, it is going to cause a corrosion of the pipeline. Even if it's a concrete pipeline or something, also there is going to be some corrosion in the top part of the pipeline. Or some also say that there is a H2SO4 buildup because of some microbial deposition in the top layer of the pipe. H2SO4 is going to build up, which is again corrosive. We know that acid is a corrosive acid. So it's going to cause the corrosion of the top layer of the pipe. Because of the top layer of the pipe is getting corroded, we call it as a crown corrosion. So this is known as a crown corrosion phenomenon. Okay. This is a very simple question. You can try answering this. I think it was a part of your lecture, so that's why I'm telling it's pretty easy. I hope crown corrosion was clear. You can now answer about patina. Okay, assuming that this is also a bit tough, let me tell you. Patina is generally deposition, right? I think that you are clear about. It's kind of a deposition over some building surfaces or some metals. So all these comes under your built microbiology topic only. So you have this patina type 1 where it is basically a film formation only. Um, Whereas in patina type 2, the surface itself will get corroded. The main difference between patina, like you have, there is some surface disturbance over the metal surface or the concrete surface of the building. So that is called as patina. In type 1, you will have film formation, whereas in type 2, you will have corrosion. So that's the general difference between patina type 1 and type 2. I think there was one particular slide which explained about different characteristic, where it happens, what is the modification that happens between patina type 1 and type 2. In your lectures, you can just go back and have a glance of it, because all these are just general topics which you should be knowing. Okay, I think we'll skip this because I told you about what emerging contaminants are. EPPR process also we have seen. Okay, now you can answer me about. I think you are clear about emerging contaminants in EVPR process. If you have any doubts, you can just put it in the chat box. We'll discuss those questions again, and you can now tell me what is a primary and secondary clarifier. I think it's a very easy question. What is a clarifier? Anyone? What is a clarifier? If you have no idea, can you just give me a thumbs up? I'll explain. Right. So we have seen about this primary, secondary, tertiary treatment now for a treatment process. Clarifiers are nothing but sedimentation tank or settling tanks. You all mean the same. A sedimentation tank, a settling tank, or a clarifier is going to mean the same. Primary clarifier means the primary sedimentation tank. Now you tell me what is a primary clarifier used for? I told you the clarifier means a sedimentation tank or a settling tank. So a primary clarifier will be used for, or a primary sedimentation tank is used for. Right from the beginning, I have told you what a primary settling tank is used for, a secondary settling tank is used for, right? Okay, just tell me if you remember those or not. Give a yes or no in the chat box. Ah, right. Okay. So settling tanks are generally used for settling of solid particles or the suspended solids basically. In the primary settling tanks, the difference is you are going to have only the non-reacted, which mean the pollutant particles uh, like the sand particles or something like that, which escape the grid chamber basically. So that particles are going to settle. Whereas in the secondary settling tank, it is particularly for the settling of the sludge or the microbial biomass that is present after the secondary treatment. Secondary treatment, you all know, we'll go for a biological treatment only mostly. So after that, your sludge and which includes the biomass will be there suspended in the water. You cannot let out the water as such, uh, stating that okay, I have put in the biomass, it has eaten all the organic matter, now there is no organic matter. No, that's not the case. Even after a secondary treatment, organic matter content in the water will be high. Why? Because there is presence of microorganisms in the water. So microorganisms also count for the organic matter. So you have to remove these microorganisms from the water. For that, what you do is you send them to a secondary clarifier or a settling tank, wherein they get settled, and the clear effluent you can take it to your tertiary system. BOD, COD are more important terms. I guess in the starting of the class itself we have discussed it elaborately. Okay, this is a very raw statement. Just tell me what is plug flow, what is CMFR, what is CSTR, and what is SBR. I'll just edit this. You just have to tell me what these individual terms mean, and then we'll be fine with. I will be fine with that. What is a CMFR? Okay, so CMFR means completely mixed flow reactor. This can be a batch system. Like most probably, you can have it as a batch system, completely mixed batch reactor. Okay, CSTR is continuously stirred tank reactor. I think that you are clear. Sequential batch reactor is what we call as SBR. I think I spoke about SBR in the last live session. And also plug flow reactor, there is no mixing in the plug flow reactor. We have a longitudinal flow in the plug flow reactor. Different configurations and different uh, reactor strategies are important in terms of process efficiency, in terms of giving a higher process efficiency, or in terms of fulfilling the process demands. So all these come into picture and have their own importance in different kinds of processes. If you if you are interested in something like a reaction engineering or a bioengineering, something like that, you can just ponder upon all these more. What does FOG stand for in the context of wastewater treatment? Completely mixed flow reactor. CMFR stands for completely mixed flow reactor. When it is a batch reactor, you call it as a CMBR, completely mixed batch reactor. In continuous case, it is CSTR, continuously stirred tank reactor. SBR is sequential batch reactor. You have any idea about FOG, fog? Right. FOG stands for fats, oil, and grease. Maybe in the petrochemical industrial wastewater and all, you will come across this term very frequently. FOG. Like whenever people talk, like they just talk in the go. Like they say FOG values this and that. You have to be aware of all these terms and short forms just to understand the context of what's being told. ARG, very simple question again. Antibiotic resistant genes. Fine. That's also a very emerging topic in the field of environmental microbiology. I think that's the last term, adaptomics. Adaptomics is basically studying all the DNA, RNA, or protein adducts in the system and determining like what effect they have, what role they have in the organism's health or metabolism basically. Adduct, I guess you know what an adduct is. Adduct is like two things directly clubbing together. For example, there is some chemical that is attached to a protein or a DNA that will cause some effect in the organism's health and metabolism, right? So studying about this, the entire sequence of all the adducts that are present in a system is known as adductomics. So with that, we come to the end of this session. I hope the session was somewhere useful for you in the course of your applied environmental microbiology course. Thanks for joining in for the session. You can just give any suggestions or comments in the chat box. I'll just, or doubts, anything in the chat box. I'll just wait for 2 minutes. You can just let me know if the session was useful, or maybe if we have to change any of this, because some information I'm just giving assuming that you will not be knowing the information, and maybe if I give a very basic explanation you will be understand able to understand it more better. That's the whole point with which I prepare for the session. You can just let me know if this can continue, or you just want only the questions, answers, and more stuff like that apart from the discussion. You can just let me know. Okay, thank you. Glad that it was useful. Others, please. Okay, if there are no comments, I would just like to remind that we'll have our next session for week 9 on next Tuesday, and you can access the recorded lectures for all these sessions and through YouTube, and the PPTs through the drive link that is shared to you in the NPTEL classroom. If there is no comments, questions, or suggestions, I think we can close today's session. Thank you all for joining in and having a very good interaction. I hope the session was somewhere useful to you. Thank you all.